Well, good afternoon, everyone. Welcome to La Jolla Institute for Immunology. I am Dr. Erica Ullman Sapphire, one of the faculty, also the president and CEO, and I am so pleased to have you here with us this afternoon for our latest installment of the Life Without Disease lecture series. So we are honored to see such a strong turnout today, and I so enjoyed getting to talk to some of you just before out in the atrium. Your presence here today with our scientists reflects something special to me, and that's a shared belief in what can happen when researchers are supported, when inquiry is sustained, and when progress is anchored in scientific rigor. This afternoon together reminds us that advancing immune-driven solutions to the world's most urgent health challenges doesn't happen in isolation in a lab bench by one person working alone. Instead, it happens in partnership with our community. So for those who may be new to LJI, let me tell you a little bit about what drives us here. We study one thing and one thing only, the human immune system, how it protects us and at times how it turns against us. That singular focus has been our North Star since our founding in 1988, when a group of visionary scientists believed that devoting an entire institute to immunology could change the trajectory of human health. That clarity of mission and that singular focus allows us to go deeper, move faster, and build insights that advance the entire field. After nearly 4 decades, that focus has clearly paid off because this little place in La Jolla is top 5 in the world in this area. We are small, but we punch way above our weight. Our 420 research, technical, and support experts work here with 200 award-winning Ph.Ds and M.Ds, and we are thrilled to have recently been named once again one of the best workplaces in San Diego. That data comes from anonymous employee surveys. In an anonymous employee survey, people can say whatever they want. We're very pleased that they find this to be an enriching place to have their research supported and work together and share ideas. And they will find, as I had, this is a really, really special place. One of the things that makes this place so special are our faculty, each one of whom leads an independent research area. About 1/3 of our faculty are among the world's most highly cited researchers. That means that their papers are in the top 0.1% of the world's scientific citations. There is no other institute that has that ratio. Harvard does not, Stanford does not, no Ivy League does not. That is very special here because of our singular focus on our extraordinary environment and the really rich technical cores that support those scientists. We attract the best and we build the best. And I'm really, really proud of that. We are also led by scientists and for scientists. So I've been the president for 4 years, and I have had the privilege of working very closely with LJI's Chief Scientific Officer, Dr. Shane Crotty. We also have an incredible 23-person board of directors led by Dr. Francois Ferret, the chair of our board. So we are built here for speed and collaboration. We don't have departmental silos. We don't have a medical school bureaucracy. My faculty would not tolerate a bureaucracy. We do, however, organize our research across 4 centers. And those are autoimmunity and inflammation, everything that hurts, cancer immunotherapy, harnessing the immune system to find and destroy individual cancer cells, sex-based differences in the immune system, interesting differences between males and females that help us understand what the mechanisms of disease are and how we can better treat and prevent disease in all people, and vaccine innovation, from how we understand how vaccines work in all the different tissues of our bodies to how we might develop better vaccines against current and new agents to come. So because the immune system touches every tissue you have and affects nearly every human disease, our faculty's work spans many of these centers. And in fact, our speaker today, Dr. Daniela Weiskopf, one of our youngest and brightest stars, will talk about research that touches 3 of these different areas. So from cancer immunotherapy to asthma, to measles, to psoriasis, to emerging pathogens, our scientists here study a pretty profound range of diseases. And that is unified by that one truth: the immune system has a hand in nearly every single one of these things. We aim not only for the betterment of human health, but to actually get beyond these diseases and imagine life without some of these things that afflict us here today. So some of our breakthroughs and progress include understanding the role of the immune system in cognitive health. Did you know the immune system played a role in Alzheimer's, Parkinson's, ALS? Developing new defenses against microbial threats, harnessing your T cells to find and destroy cancer cells, understanding how your DNA changes as we age, finding new targets in autoimmune disease, and 7 different assets in clinical trials. So there's a lot that's special that's happening right now on those lab benches. What makes all of that possible and what makes LGI exceptional is not just the discoveries, but the ecosystem that surround them. And those discoveries in that ecosystem are not possible without funding. And as you can see from this slide, LGI is extremely lean and efficient with our funding. Nearly 90% of every dollar goes toward research-related expenses. We are the leanest institute on the Mesa. You can also see that a majority of our research funding, the navy blue arc in the left pie chart, comes from federal sources, about 63% in 2024. So that federal funding, mostly from the National Institutes of Health, has long been a key foundation for medical research in America. It is why America has been known as an international leader in development of new treatments and therapies, and why Americans get those treatments and therapies first in the world. But as you've probably seen in the news this year, we have been navigating many threats to that previously stable funding. From proposed budget cuts to slashing the support for the infrastructure that we need to run the instruments and collect the data. And right now, as you know, we're also in the midst of a federal shutdown, which means grant applications are not being reviewed or awarded, and progress is again facing delays. That means patients who are depending on those next breakthroughs are stuck waiting. So with that in mind, I'd like to share a new initiative that LGI announced last week, and it's designed to safeguard discovery itself. We call it the Bridge Initiative. So before I say more, let's take a moment to watch a short video that introduces the Bridge Initiative and the vision behind it.
In medical research, the breakthroughs of today are built on decades of persistence. The discoveries of tomorrow depend on what we choose to safeguard right now. It is such a privilege to share that LGI has launched a fundraising initiative designed to safeguard discovery itself. We call it the Bridge Initiative. LGI and our nation's research enterprise are facing a significant gap right now. Shifting federal priorities, threats to scientific freedom, risk slowing progress toward better health for everyone. LGI's Bridge Initiative invites visionary donors to close that gap. There are 2 pillars: the President's Priority Fund and the Beacon Fund. The President's Priority Fund lets us invest in what is most important right now. It gives us the freedom to act in real time, to seize the fleeting opportunities that traditional funding cannot catch.
Our endowed Beacon Fund is a pillar of endurance, keeping our mission steady no matter how the currents shift beneath us. An endowment is a permanent fund that's invested to generate income for an institution year after year. It ensures that our scientists can push forward all the things we're living through.
volatile funding cycles, global crises, the long years that we know it takes to unlock the immune system secretly. Philanthropic investment fortifies both pillars. There is nothing more important in life than health. And these scientists are the ones that are very forefront in being able to create that health and sustain that health. If you would like to ensure that LGI remains at the forefront of discovery for the decades ahead and the generations that follow, we invite you to become part of this legacy.
Thank you for taking a moment to watch that. We are able to move quickly and take bold risks because of people like you, who understand that discovery doesn't happen on its own. It takes partnership, it takes foresight, it takes a shared investment to keep progress steady when the world around us shifts so much. We can't control the headwinds, we can't control the unpredictable seas, but we can build a bridge over them and build one that's strong enough to carry discovery forward. Right now, we have an opportunity to make that bridge even stronger. Thanks to the generosity of one of our board members and their family, every gift to LGI's endowment will be matched dollar for dollar until the remaining $700,000 in matching funds is met. So if you'd like to be part of that bridge initiative, please connect with me or LGI's advancement team led by Kelsey Dale. So Kelsey and team, would you mind giving a quick wave for anyone who hasn't had a chance to meet you yet? So there they are. Okay, and now it is my honor to introduce tonight's speaker, Dr. Daniela Weiskopf, Assistant Professor in LGI's Center for Vaccine Innovation and the Center for Sex-Based Differences in the Immune System. Also one of Nature's researchers on the fast track to impact. Daniela's lecture, The Immunity Puzzle, What Drives Recovery or Chronic Disease, will take us to someplace that's really interesting. The complex interplay between viral infection, inflammation, and the immune system's remarkable capacity for both healing and harm. Her lab is uncovering the immune mechanisms that explain why some people recover quickly, while others experience chronic inflammation and long-term complications from the same virus. That research is laying the foundation for next-generation vaccines and therapeutics that prevent lasting disease. So Daniela is originally from Austria, and she earned her Ph.D. in immunology from the Innsbruck Medical University before joining LJI as a postdoctoral researcher under Dr. Alessandro Sette. Her work spans mosquito-borne diseases such as dengue, Zika, Chikungunya, and she's an expert in untangling how the same virus can have very different outcomes in different people. When the COVID-19 pandemic struck, she pivoted her research swiftly, producing benchmark studies on T-cell responses that guided global vaccine strategy. Daniela's impact on the field of immunology is recognized around the world. She is ranked among the very top immunology science globally, named one of those 0.1% highly cited researchers, and she's been honored by the prestigious journal Nature as a rising star. For those familiar with LGI's Tully and Ricky family Spark Awards for Innovations in Immunology, Daniela was among the very first cohort of winners when she was a postdoc about 10 years ago. So her research and career since then reflect exactly what that Spark program was designed to do. Identify the most promising early career scientists and give them the startup support that propels their ideas and their careers to the next level. So since her Spark days, Daniela has earned multiple globally competitive honors. Today, as she deepens our understanding of how biological sex influences disease outcomes, she is not only expanding the boundaries of immunology, but also defining the future of vaccine innovation and pandemic preparedness. Please join me in welcoming Dr. Daniela Weiskopf.
Thank you. Thank you. I hope I can live up to this introduction. Thank you. So I'm very excited all of you are here because I'm excited to tell you about my lab, the focus of my lab, and why I'm so interested in immunology. Today my talk is termed Immunity Puzzle, and I'm interested in what drives recovery versus chronic disease. When I start talking to people about my lab, I always say I work on the most dangerous animal in the world. How so, you wonder? If you look at this list and you ask people what is the most dangerous animal in the world? 9 out of 10 people say oh, it has to be sharks or others. Sharks are actually on the very top of this list. As you can see, I just recently learned that more people die of taking selfies in risky locations than sharks do. As you look down the list, there's many ways to die, but the very end is the mosquito. Why so? Mosquitoes transmit one of the most deadliest diseases. So they transmit malaria, dengue, Zika, yellow fever, Chikungunya virus. Together, they are responsible for hundreds of millions of cases every year, and that makes these small mosquitoes the most deadliest animal in the world. And this is exactly what my lab is focusing on. I am interested in these viruses that are transmitted by these mosquitoes. As you heard from Erica, dengue virus, Chikungunya virus, yellow fever virus, or Zika virus, all of these viruses are transmitted by these mosquitoes. And that's what I'm interested in. And what I'm mostly interested in is this picture. This picture is true for every of these infections that I've just mentioning to you. There's always the same virus, but all of us react so different to it. Some of us don't even know. They run around asymptomatic. Actually, the ones that help transmit this virus is a lot faster. There's people that have some sort of symptoms. There's people that have more severe disease, and there's people that end up in the ICU. And if you just think back a couple of years, that's exactly what we learned through COVID, right? All of us were infected. Some of us didn't even know. Some of us ended up in the hospital. So nobody really understands why this virus is so different. It has to be in us because the virus is always the same. So that's what I'm studying. And this slide was actually my very first Spark project. So I don't know if somebody remembers. Yes. And since then, I'm still interested in exactly this question because it doesn't matter what virus you look at, the virus always has this broad range of symptoms. So I told you the most dangerous animal. That's not only what I came up with. The WHO also launches a global health initiative to actually address just this problem because we realize that these mosquitoes are traveling further and further and further, going to new areas they have never been into, and then just helping spreading this disease around the globe really. And if you don't believe me or the WHO, there's also Ryan Gosling just recently said at the Colbert show that he knows very well the most dangerous animal. He knows that the mosquitoes transmit the most animals. So believe one of us, we all say the same thing.
Not just a pretty face.
Yeah, exactly. If you go for looks, like Erica said, then listen to him. Why should you care, you wonder? And this is basically the one slide why you should care. Mosquitoes are running in your backyard. I'm sure all of you have experienced that. And this is a map of San Diego County. And if your house is in any of these red areas, the mosquitoes are in your county. The mosquitoes are right in your neighborhood. So if you look at the gray area, this is under surveillance, but they haven't detected the mosquitoes yet. But all of these red areas, these mosquitoes that transmit these viruses that I've just been telling you about are in these areas. As recent as 2 weeks ago, the last mosquito was identified in San Diego County. So I'm sure you all are aware that these mosquitoes are in your backyard, but it actually got more urgent because just a year ago, the very first cases of dengue virus have been detected in San Diego County. So these are people that never left San Diego County and came down with dengue. And as you think back of the slide I showed you, it was like for all the people that we have identified that have the virus infection, there's probably a lot more running around that don't even know it yet, because there will be asymptomatic infections. So that is just something that you keep in mind. This is why this is not a problem for like other parts of the world. This is also right here, right now. So, and that's something that like I tried to like always point out when I started working on these viruses, they were considered neglected tropical disease. They're not so much neglected no more because they are right in our front door. And this is true for like many of these other viruses that I've shown you. And today I'm picking one of these viruses, and I'm gonna tell you like what have I figured out to answer exactly that question, like what's different in people that still have symptoms and people that have recovered. And the virus I'm talking about is called Chikungunya virus. It's an African word, as you just already heard. Like this is already spread far outside of Africa. This word means like as the one who bends, because if you get infected with this virus, you have like arthritis-like symptoms, like bending your like small joints and knees and fingers. So when I started working on this virus, I was like interested, okay, what is known about this virus? And the answer is not much. We did not know what is recognized. Does our immune system see this virus? Does our immune system look any different in the people that like respond different to it? So I started looking into this at the clean slate, because not much was known so far. And I told you that like Chikungunya virus originated from Africa, but is already like around the globe. And this is the outbreaks that have recorded just this year. And you can see all of these areas have ongoing outbreaks of Chikungunya virus. So, and you can see like the major outbreak is this year is in Brazil, but like a lot of the Americas and also like parts of Asia. And if you see like very small here, France, Italy, like this has also made it to like Europe. When they had the Olympics in Paris, like there were people that were infected with Chikungunya that visited that area. So this is the same thing that we see for all of these mosquito-transmitted viruses, because the mosquitoes travel, so the virus travel with them. And just last week, the very first case of Chikungunya infection in the US, Long Island, has been discovered in a person that has never left Long Island. So the virus is here. So not yet on the West Coast, but I mean, it's a matter of time. So what am I interested? And I told you, like, I'm always interested in the pathology these viruses like induce. And as you can see here, when we get infected, all of us, there's not a lot of asymptomatic people with Chikungunya. If you have been infected, you will know, because everybody gets, most of us get symptoms onset, and that is like fever, headache, retroorbital pain, that means pain behind your eyes, rash. So almost everybody that has that, and this lasts for like 1 to 3 months. The immunity puzzle here is like 30 to 60% of the people never recover. So they have years after like infection. So we know very well when they have been infected, because there are symptoms in the acute phase. And then these people like have been infected 6 years ago, 7 years ago, and still have these symptoms. They still have arthralgia, joint pain and swelling, and they still have myalgia, so the muscle like soreness. So that was something that I was interesting. Why is that, and what is different? And that is what I'm like was interested in answering these questions. As I told you, clean slate on this virus, not much is known in humans. So are there differences in the immune phenotype between those with chronic symptoms and those that had recovered? And I looked at the cohort, and I started looking at different parameters. And you can see here, these are the donors that I've recruited. They are recruited from the Caribbean, because like 10 years ago, there was a big outbreak of Chikungunya in Colombia. These are from northern Colombia. And you can see here, if you carefully read through it, the first thing that jumps out is like 88% females. Why? So, because the mosquito bites male, the mosquitoes bite females. Like, they don't have a preference. They bite female mosquitoes, by the way, are the only ones that bite. Yeah. But they bite all the genders, both genders. And the ones that still have symptoms are like 88% females. That was the first like head scratch. I'm like, why is that? And I was looking at the symptoms at the time of infection, which is shown here in the lower part, and I was wondering, could you already tell when they were originally infected, like who will be the one that will develop symptoms or not? And the answer is no. They all look at the same in the acute infection. As you can see here on both sides, both of them have the exact same symptoms. So you will not be able to tell like, oh, you better watch out. You are most likely to like get into chronic disease. So the answer is no, you can't tell before. But clearly 6-7 years after infection, the first thing, mostly females. And you can see also when you look at the age, females in their 40s. The average age of these females was like in their 40s. So that's interesting. That's unlike many other viruses we see where like they affect mostly children or older people. Here now it hits like females in their 40s. Hits close to home for me, so I'm like, I was interested, like I better find out why that is. So how do you unravel the immune system? This is a slide that's made by Shane Crotty, like here, so I use it all the time because it just shows like what are the different parameters of the immune system that we are interested to look at. And you can see many people, vaccine developers, look at antibodies. So are there differences in antibodies in these people that have still disease or people that have recovered? Or is there a difference in cellular immunity? So my lab studies mostly T cells. That's what I'm interested in. So there's CD8 T cells, or we also call them killer T cells that help clear the virus. There's CD4 cells, also called helper T cells. These are the ones that help like the CD8 T cells that help antibodies, making like highly functional antibodies. So this is basically the three angles of the adaptive immune response that we are like studying. So I was trying to start looking into this. Okay, is there a difference in these two cohorts? The first thing I looked into was antibodies. And you can see here, always in black is shown like the antibody levels in symptomatic women. And in the red is the one that in the people that have recovered. And you can see there is no difference. Everybody has the exact same level of antibodies. So there's no difference between these groups. So, okay, I can cross this out. Antibodies do not tell you like who gets recovered and who has still a chronic disease. Okay, so what about the cellular immune response? That's the next thing I was looking at. You wonder like how are we like able to like actually ask the question to find these white blood cells that actually do recognize chikungunya virus. And I'm just going to show you like how our methods, how this is done. And we use something that is we call peptide megapools. So that means we're using peptides as small fragments that like our immune system recognizes. And we spend the entire virus, because I told you nothing is known. We did not know what of these proteins is actually recognized by virus. So we carefully went there and looked at every single possibility, like, are you recognized? Are you recognized? Are you recognized? And we answered that question by using a method that is called flow cytometry. So this is a machine that like is very well like run here. We have a great core that helps us doing these assays, and that allows us for every single cell we can ask, do you respond or do you not respond? And this is this machine that we can do that. And if you respond, how do you respond? What is the quality of your response? Because like there can also always be a difference in the magnitude. So you just see more of these, more of your cells recognize this, or the same amount of cells sees it, but recognize this response very differently. So as I told you, two flavors of T cells, the killer cells, the CD8, and the helper cells, the CD4. First question I asked, okay, is there a difference in these killer cells? And you can see the very first surprise, we don't even see there's a lot of chikungunya-specific killer T cells. As you can see here, the different proteins are shown here, and very, very, very few actually do recognize any parts of the virus. And that is not a problem of these people, because if you... oh, sorry. There's like a paddle down here. I stepped on it, as you can tell. Okay, we'll kick this to the side. So this is not a problem of these people, because if you look at a completely unrelated virus, they are very well able to make killer cells against this virus. As you can see here, CMV, cytomegalovirus, another virus that is studied a lot here in the institute. So they do recognize this virus, CD8 T cell killer cells, very well, but not chikungunya. So, no difference there, because there was no response to begin with, which is surprising, because if you study viruses, they all do usually have killer T cells that do respond. But in this case, no response, no difference. Okay, what about the last option here, CD4 T cells? Is there a difference in the helper T cells? So we did the exact same exercise. We looked carefully, protein by protein by protein by protein. What do you recognize? What do your helper T cells recognize? And is there a difference? And the answer was very surprising. In the exact same people that do not have a killer T cell response, they have plenty of helper T cells that do recognize it. You can see almost every protein of this virus is recognized. And just to compare, of course, also the unrelated virus as a control, very well recognized, as it should be. So now, is there a difference between black and red? Yes, there is. Highly significant difference between these people that still have symptoms, shown here in black, versus the ones that have recovered, shown here in red. And you can see, if you remember statistics, the more asterisks you have, the more significant it is. And you can see 4, so it's highly significant. These people, women that still have symptoms, have these helper T cells, chikungunya specific helper T cells, not any helper T cell. So we found the culprit. It is these helper T cells. So I told you the way we're thinking about this is well, is it just a matter of the magnitude? So is there more cells in these women that still have symptoms, or is it also the functional? So we have a way to study this. We can study every single cytokine they make, every effector molecule they make, and we went through many of these, but we found one that was significantly different in these women, and this is shown here. TNF alpha, tumor necrosis factor alpha. Somebody maybe that has heard about rheumatoid arthritis, may have heard this, TNF cytokine. And it is, again, highly significantly more expressed in these people that still have symptoms compared to the ones that have recovered. And that is interesting because when we think about viral infection, what we would expect is this cytokine, interferon gamma, which is usually the first forefront of cytokines that respond to viral affected cells. And as we would expect, cytomegalovirus has this cytokine. That's what we would expect from viral infected cells, but not chikungunya. So you can see there is a difference, significantly less in this chikungunya. Why? Because they make all TNF alpha. Unusual for a viral infection. So I told you we have realized first that there is many of these characteristics that points out the difference for females. So I was wondering has somebody else noticed this before? And there is studies that have exactly asked that. They ask, well, who still has symptoms years after? And others also have identified sex, female, and the years of age, average age of 45 years. So this is not only in this cohort, this seems to be something that is universal. So this is, if you have been infected with the virus, you're female and you're in your 40s, you're much more likely to develop this chronic disease. So my part is trying to find out why that is. So we have found some small answers, not all, but we have identified that the sex is really the one that makes a difference in this chronic disease population. I showed you that there is higher frequency of these chikungunya-specific CD4 T cells in this chronic population, and TNF alpha is the cytokine they mostly reproduce. So I'm an infectious disease immunologist, but I was thinking, well, when I was in grad school, this looks awfully like something that I have seen before, and this is what we usually see in autoimmune diseases. So I looked into this and I found what is the autoimmune disease that looks much closely to what I see here, and that is rheumatoid arthritis. The clinical symptoms are similar. Both of them have like this pain in the wrist, small joints such as fingers and wrists, have like these symptoms years after. You have fatigue, myalgia, insomnia, and depression. Both these chikungunya-like infected women report that, and also like the rheumatoid arthritis patients. The cytokine profile is very similar. I told you, like, I was very surprised to see all this TNF-alpha in these chikungunya-infected cells, but so is exactly the same that they see in these rheumatoid arthritis. And we know that also, like, rheumatoid arthritis primarily affects middle-aged women. 60 to 80% are women. So there's a lot of similarities, even though they come from completely different angles. Both times we don't know what actually like causes the disease. And we know that there's anti-rheumatic drugs are effective in some cases of like the chikungunya infected people, but really it's not like widely given, because like right now, if you are infected in these like countries, like these women get painkillers. That's it. So I think there's some usefulness to look into that, if that could help. Because like, so I convinced you that like, very similar, now this is a viral infection, very similar to rheumatoid arthritis, to autoimmune disease. And this is not the first time we all heard about like a possibility of a virus triggering like an autoimmune like disease. We all know somebody that has long COVID. There's another virus that triggers like, possibly triggers like these symptoms. We all recently heard that like possibly infections with Epstein-Barr virus can trigger multiple sclerosis. So I think the thinking of like having like viruses that can trigger any of these autoimmune diseases is much bigger than like what we already know. So this is something that we need to like look into more detail. And I told you like this viral infection, more women, and this is also like and the similarity to autoimmune disease. And there is many autoimmune disease that actually follow exactly the same pattern. So many of these autoimmune diseases have more females that are covered from that, and as you can see here down the list, that is something that like follows it. And that is something that like I start to thinking about like, well, does anybody have ever looked at the viral triggers for any of these other like autoimmune diseases? That is something that I will follow up on, and I will keep looking into it, because it's possible that this is very much closely related. So if you are interested in this, some of you are like scientists themselves, like this has all been published. This is basically showing that for the first time that there is a difference in these chronic infected people, with, and that can be read up here. And with that, I like to show you the acknowledgement. So this is my lab. You heard from Erica about the SPARK program. There is 4 SPARK awardees on this picture. So half of my lab has won one of these awards. So that just shows how impactful, like if you have donated like that is, because they really have all been like, went through the same thing. And you get confidence if you win this award. You learn how to talk to like non-scientist people, and you learn how to think through a project. So I will probe Barbara if she knows who of the 4 here have the won this award. And of course I have lots of collaborators, like all the people in my lab that you see in the picture and here on the side. The ones in blue are the ones that like mostly were contributing to this work. Alexetta and Alba Grifoni's lab have been heavily involved in this study as well. There's people that like help us with the antibodies from the University of North Carolina. They're listed here. And then as you can see here, Universidad del Norte are the ones that helped us collecting these patient samples. And with that, I hope I like ignited the curiosity, like so you can see and support us and follow us in like if we can figure out like all these different immunity puzzles. Thank you, and I'm happy to take questions.
Thank you, Daniela. What I've always liked about your research is it's so thought-provoking. You know, we have these paradigms that virus bad, immune system good, but then the immune system in these cases starts that fire that never goes out.
Yeah. This whole project was always like, this is not in the textbook. This is different. Like, why no CD8 T cells, no killer cells against viral infection? I didn't expect that. Why do not making interferon gamma? That's what we expect from viral infections. No, they're making TNF alpha. So that's why I'm saying all of a sudden I'm realizing this is different than what I'm usually thinking about viral infections. As I said, I'm an infectious disease immunologist. And that's also like you said this in your introduction, like, you created all of these centers here. So now I'm thinking, am I dealing with like an autoimmune disease? I can go here to the next floor and ask, because now you created like all these centers like that have the specialists now. So that was very helpful, because I was like, I don't know, I need to ask. And I could because of that.
Thank you.
Yeah.
One of the other things I think is most interesting about your work is, one of the things we grow up learning about is the one size fits all approach for medicine, that we dose things by body weight, and what's good for the goose is good for the gander. But your research kind of suggests that it's not. And I know from your work and some of the work of your trainees that goes way beyond this one project. So what are some of the other things on your lab bench that address that difference?
Yeah, so we are looking into the same question about dengue virus. And as I said, like my very first Spark project tried to answer this exact same question. Well, like, how do I know like you have been infected, and how do I know that you will most likely end up in the hospital? I would like to know if I have infected. So that's something. The other project that like goes more into like the sex difference, and is something that like is really run by Paro, another Spark awardee, and she really has asked the question, well, menstrual effluent, half of our population has it every month. So can we actually find anything that could predict like, any of these diseases? And she has established a protocol that actually is able to look into this. Like we can find T cells in menstrual effluent. Very good. Like it's non-invasive. You just like drop it off. And you don't have to do a biopsy like to find out if you have endometriosis or possibly get an endometriosis. So that is something that is like really like mind-boggling to me. Like, why did nobody look into this before? So yes, yeah. So.
Yes, sir.
Give me just 1 second so we can all hear you.
Sure. For both CD4 and TNF, there's a range for each, which to me certainly says that there are other causative reasons for what you're observing, which I don't want to get into necessarily. But what the question I will ask is whether you've done a correlation between high and low CD4 and TNF, and whether those correlate at all.
Yeah. So if you have more CD4 T cells, if you have more TNF alpha, that's the question? Yes, you have. Yes.
No, in these patients
Yeah.
where you see high levels, yeah.
Yes, it is. And the one other thing that like I'm starting getting really interested in is exactly like your question. Like, so we noticed when studying this, like that not only the CD4 T cells making TNF alpha, all of a sudden we also see like, like macrophages, monocytes making in these patients, making a lot more TNF alpha. So that's something that we are actively looking at right now. Like, is that possibly like why there's this connection of these like helper cells that make TNF alpha, like because they get like primed by like antigen presenting cells that do make a lot of TNF alpha, and that is something that we're looking into right now. Yeah.
Actually, one other thing that came up that you didn't present is whether there's persistent viral infection in any of these patients, and whether that makes a difference.
That was my first question too, and I asked every virologist, like, is this virus still present in these people? Because that would explain why they still have the T cells, and all virologists say no. When we look into the plasma, we cannot see virus. So either this virus is present and goes somewhere where we cannot detect it, because you cannot do biopsies from like all these immune privileged sites.
But this is 6 years later. I'm wondering if the initial course of infection is any different in the patients.
No, the initial course is exactly the same. That's why I'm saying they have the same symptoms, they have the same viral loads, they have the same like, that's the other question I had. Is the virus still there? And then what happened in your childhood? Like, so what happened when you were infected? Same, it's the same. It's just later that comes up, yeah. That's why one of these project answers more, like, asks more questions than answers.
I have two questions. One is trivial and small. The other one is very large. The small question is, on your slides, the N's were often less than 20.
Yes.
I wonder why such small N's.
we have recruited like close to 200 of these patients, but we already started to see this like stark difference with very little people. So we were like, this is, so that's when we started looking into this other reason. So you're absolutely right. We can add more donors, we can add more ends. We have many more samples to answer exactly that question. But because we saw so quickly like the difference, dramatic difference, we started looking into like what causes this.
The big question is, what is the pattern, or how far away are we from treatment for this virus?
Yeah, that's a very good question. And UCSD, together with George Washington University, actually runs a clinical trial in this area, and they give these people methotrexate. Does it work or not? We don't know yet. We all are still blinded. So, but yeah, that is a good question, because that's the other question I asked. Is this a virus, and like, can these women get TNF-alpha blockers? And they were saying, their answer I got was like, this is like way too expensive. So, in these areas. So, we will see. I know what I ask if I got chikungunya. Yeah.
Thank you very much. I'm interested in the mechanics of how you recruit patients into these studies and how you translate the results into clinical practice on an accelerated basis.
Yeah, so the recruitment of these patients was fairly easy because this is also in the US a notifiable disease. So every doctor has to report them to the health ministry. So we just asked them, like, can you call them back? Because we asked them like 6-7 years later, right? So yeah, so they had a record of everybody that was infected, and like asked them if you want to come back to donate blood. So that's how I found these people. The downside of this is like, now people ask me like, well, have these women had pregnancies? Like, I don't know, like, did they have 1, 2, 3, 4 children? I don't know. Like, you know, what is their hormone levels? I don't know, because they have not been recruited for this exact question. But that is something that we're trying to hope now, together with UCSD, to answer that, because they have a much more carefully recruited cohort. Yeah.
Thank you.
So another question, Dean?
Yeah, I assume the CD4 levels in women may be related to hormones, because it seems that postmenopausally they're not having it. Do you have any ideas of why this happens? Why whether it's estrogen, progesterone, or something else would change the way that you're putting out CD4?
Yes, that's a very good question. And that is something, as I said, because our cohort has never been recruited to answer these questions, because right now I would give like a completely different questionnaire, like asking exactly that question, like, can you donate like a plasma so we can measure your hormone levels? Can you tell us like if you had pregnancies? Can you tell us like, and a lot more questions. So yeah, you're absolutely right. This was not what we expected when we started looking at this, but now we have a lot more questions. So yes, exactly that. Yeah, good question. Thank you.
Do we have time for one more, I think?
Yeah.
I'll try.
I can talk about chikungunya
Okay, so
all
after coming
night.
to these
so yeah,
lectures for
you're
many
good.
years, I'm
You
as
let me-
confused as I ever was. Could a viral infection make a change in somebody's genes and thus trigger this anomalous response?
We know about viruses that can do that. We don't know that about this one yet. But also, I think in this case, nobody has looked yet. So I don't know yet. Yeah, yeah.
I'll write a novel about it.
I'll take the last question. If you had unlimited resources, what would be a dream project you would like to do?
Well, that's why I also talked about these projects that I'm interested in. One is like what happens on the other side of the immune system. Like, so what happens to the macrophages, monocytes? Well, why is there all this TNF-alpha like also made there? That would be a project I would be very interested in. What triggers these CD4 T cells? Is it like something, a circle that goes around, like connecting the adaptive and the antigen presenting cells? And the other project is like something that, like I also mentioned, like the stems from Ampara's Spark project. I would really want to know like what is in these menstrual effluents, because that's a very non-invasive method to sample like women's health. So that would be the other project I would follow up with. Yeah.
Thank you.
Oh, you're here. Okay.
I'm here. So thank you, Daniela. Your research. What I think is really important about your research is not just that population in Colombia, but that phenomenon is seen so often in this country. So many people debilitated by long COVID, by Lyme that never goes away, by a virus that I don't even remember having 20 years ago, that we don't know what it is anymore because the smoking gun's gone, but their life and their career and their health are debilitated by an immune response that will not turn off. And there's just so much to learn there. And it's just so thought-provoking. So thank you. And to all of you, everyone gathered here today, thank you for your time, your curiosity, and your partnership. So that progress in science like Daniela's doesn't happen in isolation. It takes a whole community of scientists, partners, patients, clinicians, advocates, funders, community stakeholders, all of us working together toward a life without disease. So as you can see on this slide, there's a few ways you can become more involved. I invite you to consider joining the Bridge Initiative, ensuring that research like Daniela's continues to advance now and for years to come. 2025 may be an especially strategic year tax-wise. The federal tax changes for 2026 will limit some of your deductions. 2025, I believe you can deduct every dollar you donate. So if you'd like to try to navigate that, Kelsey and her team can help you figure out how to navigate that process and connect you with the most impactful philanthropic opportunities at a giving level that feels right for you. And the kind of research you heard about this afternoon on emerging threats that we know are coming, now Long Island, now San Diego, that depends on flexible, reliable funding that's available when the opportunity strikes, and when traditional grants fall short or would take a year to come in. So that Bridge Initiative also makes that rapid response possible, ensuring that promising ideas like from Daniela and from her lab can move forward uninterrupted. So your generosity fuels that momentum. It gives scientists time, it gives them stability, and it gives them the freedom to ask daring questions that lead to life-saving solutions. So now, as we're going to move the reception into the atrium, I invite you to continue these conversations and connect with our scientists. So thank you for being part of Life Without Disease. Thank you, Daniela.